Start the journey of ERPs, the event-related potential, and the ERP lab. So myself, Pragati Gupta, and I have completed my master's from Gujarat Forensic Sciences University, India, MSc Neuropsychology, and I'm currently searching for PhD options in the field of cognitive neuroscience. So yeah, I'll just share my screen. Hope you can see the slides. If you guys have any question, you can put it in the chat box and you can later take the questions. We'll start. So this is a short introduction of what are ERPs and how is it being used as a tool in the MATLAB interface as a ERPLAB toolbox. So we'll deep dive into the world of ERPs, and I'm really thankful of ScienceBeam for providing me this opportunity to explore this area and give a presentation. So these are the topics we will be covering, like the basics of event-related potential, the electrical signals generation, 1020 system re-referencing impedance, the various ERP components, early and late ERPs, in the domain of research and clinical field. We have the key points as in what are the important facts when we are dealing with ERPs, the difference wave measurement windows, filtering, pre-processing, etc. What are the ERP amplitudes and latencies present, how they are being studied in terms of statistics. We'll be doing that a little bit. Then we have a short introduction on ERP lab, and then finally we have advantages and disadvantages. So in between we will have a short break where we'll be showing you a short video on the ERPs and introduction of what the ScienceBeam is doing currently. So we'll have as a break. So let's start. Firstly, before ERPs, we need to know what is EEG. So EEG are like electrographic signatures of the electrical currents in the brain. And with the help of EEG, we derive those ERPs. So here in this first slide, we are seeing what are the methods used for observing the living brain. So in the y-axis, we are seeing it's log size. So log size here are like kind of a spatial resolution. So, and in the x-axis, we have log time. That is the temporal resolution, which means these brain imaging or physiological methods, brain imaging methods are being looked in terms of the temporal and spatial resolution. For example, in terms of EEG, we are seeing that the area which it is being captured is like the brain, from the cortex, we are capturing those EEG signals. But for the example, if you see the patch clamp technique or the single unit or multi-unit recording in animals, so from there we're seeing that it goes deeper into the cortex, like it records electrical signals or brain signals from like dendrites or neurons. Here we are seeing particularly our EEG, which is like with a higher temporal resolution and little like difficult to measure the spatial resolution. So we have EEG and MEG in the brain part, and we have other techniques like fMRI, which have the which takes the brain area from the blood oxygenated dependent level, and we have PET imaging technique, lesions, micro lesions, TMS. All these are like going deeper into the cortex rather than from the surface of the cortex, from where we record the electrical encephalogram. So we'll go next. So here we're trying to see how and where it is measured. This is EEG. So we are starting with EEG firstly. So which means like what are the recent technological advancement in EEG? So firstly, on this left side, we are seeing this 9-electrode recording device from Japan in the early 1950s, from where we used to record EEG. Then we have this David Fedner, was the propounder, like initial propounder in the interpretation of EEG signals. So he's wearing that cap. This is like I'll show you the pointer is not, sorry. So here the third diagram from the 1980s, like showing high quality EEG caps. And when we are moving forward to like the high density EEG recordings and like low cost resolution. Now we in the late 2019 or 2020, we are seeing like the BCI, brain computer interface, where we are using like we don't use like the amplifiers and all in terms of like uh, leaving the EEG part, which means we're using both EEG and ERP as a technique here. And here down we are seeing, for example, that four of the participants are from four of the participants we are recording the electrical signals at the same time. This is known as the hyperscanning. And we can also record EEG from like infants. We can also provide EEG from the neurofeedback, like the real-time feedback we can get from this type of technique. And this was the earliest way from which we used to record the EEG in terms like from the paper recording. And also with recent advancement, we have different equipments, different techniques. Okay, let me just mute myself, please. Unmute yourself. Yeah. So yeah. So the recent techniques like we are using in EEG, like topographical analysis, different signal processing methods, source localization, single trial analysis. So these are recent advancements in EEG. So we are looking today at the event-related potentials of it. So next we move forward. So how are these electrical signals generated? So it might seem we have all studied the basic how does action potential works. So it's a different generation of electrical signals. It's not from action potential. We have postsynaptic potential working here in EEG. So in the neuron, from the neuron, the information is carried to the axons. Then it releases the neurotransmitters. And here, the dendrites receive these neurotransmitters. Later, the axons of other neurons causes polarity changes inside the neuron. Then further, we get those signals from their postsynaptic potentials of the neuron, and specifically we use the pyramidal cells. So why pyramidal cells are used in the generation for event-related potentials, we will be seeing. So this is the structure of a neuron, you can know. So here basically we are trying to study that this is a stellate neuron and this is a pyramidal neuron. So why these two kinds of neurons? Specifically, we use pyramidal neurons in studying. So the pyramidal neurons are used in the generation of ERPs because of the dipole orientation and the structure of the pyramidal neurons. Because in the stellate neuron or other type of neurons, the random orientation of these dendritic structures cancels out the dipoles of the voltages we observe from the scalp regions. These are cancelled out. So, but in the pyramidal cells, we have a apical dendrite towards the surface of the cortex, and we have the basal dendrite towards the deeper underlying the cortex near the white matter. So here, these orientations, when there are like hundreds or billions of neurons activating at the same time, so these creates like voltages. These creates a dipole, which in turn, in summation, provides those small electrical signals which we observe from the scalp regions. So that's why we use pyramidal neurons. I mean, like pyramidal neurons are being the key propounder for generation of these event-related potentials and electroencephalogram. So this is a short explanation of like how it does. So for example, we have this dipole, and these are the pyramidal neurons which are like parallelly oriented, and we have the equivalent current dipoles. So equivalent current dipoles are like the average of all the short single dipoles in the neuron. So here we get those averages from the scalp regions. So if you're seeing the head of the arrow, which is positive, which means like on the apical dendrite on the cortex, the generation of signal are negative. And deeper layer of the cortex, near the white matter, there is a positive. positive electrical signals, which in turn indicates that this is equivalent current dipole is being generated when there are like excitatory neuron. So this is a explanation of the excitatory neuron. When there's an excitatory neurotransmitter is present, we see this type of configuration. And we can see the cortical pyramidal neurons are parallel to each other, allowing their dipoles to sum to a large value. So these similar orientation of the cortical pyramidal neurons act as a feedback, and it provides a mechanism for the signals to pass on forward to the cortex, and we receive those signals. Rather, in the right hand side, we are seeing the random orientation of these neurons. In a similar way, the extracellular electrical fields produced during the neurotransmission sum across the neurons in an area. So these equivalent current dipoles, the summation of these small dipoles in the neuron, it is like acts as a perpendicular to the cortical surface, specifically of the pyramidal neuron. But the same way it is observed in the scalp region, it's not very easy to understand the underlying cortical regions, because the area of the brain or the region of the cortex are like folded in a different manner, like very complex manner. So in the orientation of the sulci and gyri, the folds, which like blocks or like which provides a different route of electrical signals, the flow of information, the way we observe those signals from the scalp. So this will give us a negativity in our scalp. So this EEG electrodes, for example, we are seeing that on the one side, it has a positive orientation, positive one side of the dipole, it's a positive orientation, and on the other side, it's a negative. So like when we see electrical signals from the brain, so there are multiple dipoles. It's not a one single dipole. It's like multiple dipoles of different orientations we observe, and we see those small waves. Now, basically, how we acquire the data. So we have the EEG cap on the participant in the first image. Then we place some gel for the conductivity of the signals. Then those signals are being transformed to the amplifier, which then amplifies the small electrical signals. And then we acquire those in the data acquisition system. It's shown very simply, but we'll explore more detail later. And this is a like short explanation of how do we take, how do we place those electrodes. So most of the EEG systems already have these electrodes placed on the cap. So they have a defined internationally these placements, so that it's easy for comparison. So these are placements of different electrodes. So here these are the Nasian and the Enian. So Nasian is the region between our forehead and the nose, like this area, and the Enian is the like back part of your occipital region, which is like a small bump in the back region. You can feel that. So this is the Enian. So from here to here, the basic circumferences we measure, and we can take the midpoint, which is the CZ. So from here we observe different electrode points. We take different percentages, like 20% of 2C4, 54. So this way, like we have 10, 20 percentages of different electrode sites. And these name, like for example, the F, T, C, P, and O. So these represents the lobes, for example, frontal area, the central. Central is for like, because in the central midbrain region it's there, that's why. The P is for parietal, T is temporal, occipital. And the odd numbers are in the left hemisphere, and the right number on the even numbers are on the right hemisphere. And the region and the electrode closer to the midline are like closer, it's like near to the 1 to 5 range of numerically. For example, FP2 is near to FPZ, and for example, F7 is like far away from FZ. So that's why it's because like it's a little far from the midline region. So this way it is being derived. This is the 1020 electrode system, which is like internationally followed to record the EEG and like how to place the cap. Then we have the impedance. So impedance is basically like how good the signal is, like when we start the recording. So before starting the recording, we need to take the impedance, which means like how good the contact is of the scalp and the electrode. So here on the left, we are seeing like impedance chart of a 128 electrode EGI system, which is like we need to keep a range in which we are seeing that it is a good contact with the scalp. For example, in the blue, 0 to 50 kilo ohms is assigned as a good impedance, like low impedance. So we are seeing most of the regions have a good contact with the scalp. So this way we identify like how good the contact is, so that we don't have a large amount of artifacts and noise in our data later for processing and understanding the signals. In a similar way here, like these are the brain products, ActiChamp caps and electrodes. Here, like with the help of gel, when we like put the gel inside the electrodes, it automatically like it has a preamplifier inserted in it beforehand, and it automatically identifies that it has a good connection or not. So here this green, it means it has a good connection with the scalp, and the range of impedance is good, like it has below 0 to 50, means it's within 0 to 50 kilo ohms, rather in the red one, which is like not yet connected, so it's showing red, so it has a preamplifier. Rather on some systems we need to like check with help of chart, like is it like how much the impedance is. So before starting the recording, we require these impedance so for like further examination. For example, here 10 is not connected nicely, so we need to like put more gel or put more salt solution to make it blue or to make it in a green region. Similarly, the electrode impedance, in short, is the extent to which the flow of current between the electrode pellet and the living skin is impeded by the layer of dead skin cells and oils on the surface of the skin, which means like the dead skin cells on the scalp also, it's a low conductor, produces low conductivity. So for some systems, we need to abrade the like clear out the dead skin cells with the help of like blunt needles, so that the impedance is low for like good conductivity of signals, good conduction of signals. So here we are seeing like with the help of, this is the electrode pellet and this is the electrode gel, which is like getting good contact with the surface of the scalp, the epidermis, and this is the abrasion, and we abrade so that there's a high contact of the gel with the scalp. And for example, if there are sweat on the surface of the head, so it can act as a like noise, like low frequency noise, so which affects a lot in the interpretation of EEG and ERPs. We'll see later. So I was telling, these are the preamplifiers which are there in the brain products, ActiChamp. So in a typical laboratory system, the outputs of the electrodes are sent to a device that filters and amplifies the voltages and then turns the analog voltage into a discrete digital values. So the signals we receive from the brain are like in analog voltages, and these are being transformed into the digital value. So AC to DC transformation, which is done through ADC converter, analog to digital converter. But these were like done, ADC converters were used before, and recently we have a amplifier which does all these ADC conversion. So we have two rooms in an experimental setup where we like where the participant sits in one room and the other is like the data acquisition room, and we sent on the event codes to the stimulus computer to the where the participant sees the stimulus, and we observe those acquisition, the signal acquisition. Next, after having the basic recording, we also have the reference, which is very important part while recording EEG. Why it is very important, we'll see, like here you can see these are marked as red, CZ, EM, these are the mastoids region. So reference is basically the summation of the activity, like how like from which region will be getting, we are minusing those voltages. So these EEG are being acquired from the voltages we observe. So voltage here is measured between active and ground. So in this diagram, we'll see like voltage is measured between active and ground, that is A minus G, and voltage is measured between reference and ground. So basically, when we record an EEG, we have active electrodes, a ground, and a reference. So these are active electrodes, FZ, CZ, PZ, and these are ground. One is ground, one is reference. So basically, how it is important, like the voltage is measured between the active and ground. Firstly, the active electrode is subtracted from the ground. Then the active electrode is subtracted from the reference. So the output is like the ground is cancelled and we get the active minus reference. So that is why like the active minus reference is the voltage between these two points, that is active and reference, which means like any activity we are observing from those regions, like from those underlying neural generators are the activities from the active and the reference electrodes. So any distortion in the reference will be distorting the signals. So the reference location is very important. So here, like we have, so basically we want a very neutral, like relatively neutral activity from which means there is no activity in the surrounding regions from where we place the reference. So that is why in EEG you usually have like the CZ, average of the mastoids, average of all electrodes, but basically all of them have some amount of neural activity. So we are trying to, so what we do, like we usually choose those references which are like previously used in researches. So we use, for example, if we are taking one component, we are studying one component. So we see that previous research, previous how they, what reference sites they've used, what products they've used. So obviously, was saying that like in an ideal world, we would want a reference electrode somewhere on the zero line. So here, where there are like there is no signal present, so we would know the, for this to know the zero line to be known for us, like we need to understand the location and the orientation of the dipoles, generator dipoles. But as we were saying that there are multiple dipoles, multiple underlying components, so it's very difficult to find this zero line. So that is why in practice there is no such electrically neutral place in the cortex from where we can place those reference electrode. So bottom line is there is no matter where the reference is located, is picking up the real signal that is then inverted via the referencing process. So basically when we are starting with the EEG, we need to understand the importance of reference, like where we need to place the reference, the impedance. So these are the basics which we like need to delve deeper into it. Then these were some of the basics. Now we move a little forward, like brain rhythms or neural oscillations. As we do, we have like 5 types of brain rhythms, like the D tap G we say in short, just like the delta, theta, alpha, beta, and gamma, D tap G. So starting from the slowest frequency to the highest frequency, we have variability in the cognitive function also. Like the delta wave is like from the deep sleep, usually seen in the non-REM sleep, in the abnormal awake state. And for example, theta activities is usually see when we have participant meditating, like focus meditation, like increased mental workload, high mental workload task, like working memory, memory consolidation, alpha activity when eyes are closed, like sometimes also seen in the cortical plasticity in motor cortex in neurorehabilitation specifically. In the beta activity, we see like when there are like higher cognitive processing are like associated with mental motor imageries and related to sensory motor cortex. In gamma activity, we have like the cortical binding, visual perception, and microsaccades. So these are like, so when we move from the higher frequency to the lower frequencies, we see that uh, there is a change in the way we look at. I mean, like, for example, in the delta activity, it's like slow oscillations. Slow oscillations will travel down in the cortex, like from one region to like it is a farther connection. It travels long route. It takes an information from larger region. Rather than the gamma activity, it is like focused. For example, gamma activity, if we see in like the occipital region, so it has a very high uh, like short amplitude and higher frequency. So like the lot of neurons are activated in this specific region, that is the occipital cortex. rather than in the delta region. For example, it is traveling down from the occipital to the frontal region via passing through various phase of neuron. Then similarly, some more functions of brain rhythms and cognition, like from navigation, so basic cognitive functions to higher cognitive functions and basic sensory perception too. So these are some of the functions of the neural oscillations. We can have deeper. So now basically the motive of the study was try to study what are ERPs. So the raw EEG, when we see, we cannot determine like what are the specific cognitive states. So for that we require like specific processing, specific time related events to process those information. So the raw EEG can provide our understanding of these brain rhythms and cognition of these brain rhythms used in sleep researches, used in clinical population like epilepsy, dementia, or Alzheimer's. But if you want to study in more like in terms of stimulus-based or response-based task, in terms of neurophysiology, we used event-related potential. So the raw EEG cannot indicate the general state. That it can indicate the general state, but for understanding the specific neurocognitive process like affect or emotion or some like motor tasks, we require those specific processing. So these are like event-related potential, as in like, so these are the epochs, like these are four epochs, and these are the stimulus and the response. These are the markers. So this is at this time point we see a stimulus and then we respond to it. So these are like event-based recording. So to pull out an event-related activity, an event code is included in the EEG file, indicating the time and identity of each event. So these are markers which are being placed before the recording starts, as in like while designing the experiment, we put these markers simultaneously while we put down the stimuli. Also, if you want to like when the recording is live, you can also put markers like if we see some different activity. Well, for example, if the participant is dozing off or like making a specific movement, like specific artifact like jaw clench or like moving or something. So physically event-related potential and electrical potential generated by the brain that is related to an event. So any event in terms of stimulus or response. So the average of all these we see using this event-related potential. So here we see different components, and it is indicating a different cognitive functions of it. So first we'll see what is P and N. So P is like positive, P stands for positive and N is for negative. So this is the polarity. So P is for like the positive going peak, and 1, 2, these are like the ordinal positions in the waveform. If number is like less than 5, it means that it is based on the waveform. That is like it's based on the ordinal position on the waveform. If it is greater than 5, it means it is in the latency. Sorry. So for example, P1 is the first component, first positive component in the ERP, which is indicating the initial sensory response, a higher perceptual process. So these are the early components. So these are used for the sensory processing. Then we have the later components like P3, N400, P600. So these are the later components which are specifically used for the higher cognitive functions like working memory encoding, emotional responses, arousal, etc. So, and also we have here the pre-stimulus activity, like the pre-baseline measures, which is very important, we'll see. And here the preparatory processes can be seen, like before the stimulus comes, what are the specific cognitive function? What is that? What does that mean? Like we can also look through ERP. And specifically I was saying like, so this is like stimulus logged events. Like we are segmenting from the continuous EEG, we are segmenting the stimulus logged events, like stimulus 1, stimulus 2, stimulus N. And for one participant, we are getting all those stimulus. For example, if we have 60 stimulus, we are getting those 60 segments, and then we are averaging it, and we are seeing those peaks. Seems easy, but the underlying assumptions and inferences we draw are a little difficult, and we'll see later. And these are the stimulus onset, and they're showing the latency. And here, like in the x-axis, we have the time, y-axis, we have the voltages in microvolts. We see the early and the late components. Specifically, yeah, these are the EEG. We are seeing the event codes, and we are seeing the response and the stimulus. And this way we analyze, and this is a recording room like kind of situation. The participant sits with the computer and have the stimulus, and it button presses, and we see these markers and the continuous EEG. So the two ways which kind of we use in the event related potential are like the stimulus logged averages and the response logged average. The stimulus logged average is like the activity of the participant when the stimulus was present, and when the response, when he performed the response. So we see the mean response time and the mean stimulus time. So this way we observe those activities in the terms of amplitude and latencies. We'll see. So now we'll move on to the ERP components. So these are some of the ERP components. We'll go briefly through it. So we'll start off. So some of them are like the sensory components, P300 and 170 for like face perception, or like specific to the expert, N400 for language processing, P600 also for language processing, error letter negativity, feedback letter negativity. So we have all these. So we'll see slowly how it is generated. So firstly, we have the C1 wave. So as you have noticed that I've not told like what the C means. So initially in this slide, we had started from the P1 and the positive going and negative going, but C is like the first wave which we see before P1 also. So these are like generated from the primary visual cortex, and these are the cross connections in general, like when we the connections we have see in the motor areas, so and the visual cortex. So the upper field has the negative orientation, negative polarity, and the lower visual field has the positive orientation. So when the stimulus is in the upper visual field, we see a negative deflection. So if we see a negative deflection, then only we can distinguish the C1 from P1. If there is a lower visual field stimulus, and then there will be a positive polarity, which is be like overlapping with the P1 wave. So when there is a negative, when there is a stimulus in the upper visual field, then only we can see the C1 specifically. So this is because of the orientation of the calcurrent fissure. The way it is folded up, so the upper and the lower visual fields are opposite to each other. That is why this crisscross happens, and the lower and the upper visual fields is being seen like opposite in polarity in terms of ERP waveform. So this is the first component, the C1 wave. Then we'll see the P1 and the N1. So P1 and N1 are usually we see for the attention task, and specifically the special attention. And these are the early sensory components, which means that these are being affected. These are affected by the experience of the participant, the stimulus, the physical properties, specifically the physical properties of the objects, so the stimulus. And for example, the P1 and N1 are larger for attended location stimuli than for ignored location stimuli. So if we see this task, so if the object is in the left side of the screen, so this means like the attended area will be showing a larger P1 and N1 rather than the unattended area, but it's the ignored area. So this way, like we see like how much the person is paying attention in terms of cognitive function, we can see. Then we have, for example, in a stimulus condition, in a task, a similar task, for example, if we are giving two objects and we are showing them in different light conditions, and we have asked them to pay attention, we can see the bright light condition have a higher P1 N1 rather than in the dim light, because like attention is greater there. So these, the early components, the C1, P1, and N1 waves they're highly sensitive to the physical properties of the stimulus. That is why it's very important to see these waves first to understand the underlying cognitive deficit or like underlying cognitive dimensions rather than going to the later components. Then also like visual ERP and auditory ERPs are different. You can see the N1 and P1 in terms of visual ERPs are different, but visual P3 and the auditory P3 are similar. This is a classic N170 ERP component and the paradigm. So here, for example, the participant is being shown with the help of fixation point and the stimulus faces and the car. So these two objects, faces and cars, are being shown, and we are seeing the averaged ERPs of these two objects. So when we see in the faces, there's a negative polarity in around 170 milliseconds, and there's a greater amplitude for it. So which means like greater amount of resources and faster processing is done for faces rather than for cars. So this is for N170 ERP component. And then we have a typical oddball paradigm, which means like there are two types of stimulus. There's a rare stimulus and there is a standard stimulus. So standard stimulus are like, for example, here are X. So in these are 10 stimulus. From there, we have 80% of X and 20% of O's, which means like 8 of them are X and 2 of them are O's. So we are seeing trial by trial the EEG of it, and we are seeing a consistent positive deflection at around this time point, around 300 to 500 milliseconds, around all the trials. So when we do an averaging, we see a positive wave and at around 300 milliseconds. This is an average of 8 oddball trials. But for getting a better ERP component in terms of P3, we require more trials so that we cancel out the noise and get the actual component. Then we have the MMN, the mismatch negativity, which are like usually the auditory mismatch negativity. So here what happens similar to oddball, here what happens like these are a kind of tones with different frequencies. So here, for example, 80% of the tones are of 1000 hertz and 20% of the tones are of 1500 hertz. So when there are 20% of, when there's a presence of 1500 hertz tone, there is a like a greater negative component at around 100 or 200 milliseconds, which is like indicating that it's like kind of a sudden attention towards the task, sudden attention towards the object, and it does not require subject to perform any task. If the person is reading a book, he or she might get this greater activity, greater negative component around 200 milliseconds for deviant stimulus. That is for the 15 hertz. So this is why, that is why we'll see like how this is being used in the clinical setup, this ERP component. Yeah, so similar like for the FZ electrode, we are seeing the MMN in terms of the standard and deviant. Standard means like the actual 80% stimulus, that is 1000 hertz, and 20% is the deviant, 1500 hertz. But here the deviants are different in terms of frequencies. We have different stimulus, and we are seeing the difference wave. So we'll see what are difference wave actually the later half. So the mismatch negativity is being isolated by the difference in which the standard stimulus is subtracted from the deviant stimulus, and we see the actual ERP waveform by the subtracting the frequent minus standard, frequent minus rare stimulus. And these are the cortical generators. For example, near the frontal medullary region, we are seeing the activation of the MMN and mismatch negativity. And we are seeing in ERP and the scalp maps, the ERP standards and ERP deviants, and the frequent deviant minus standard difference wave. Next is the N2PC component. This is the posterior contralateral component, which means like, for example, if we are seeing on the left side of the visual field, there is a negative deflection, negative going voltage at the right side of the right hemisphere because it is contralaterally being attended. It is like contralateral observation. So this is a negative going peak over the posterior electrode site when we observe any stimulus and when the object is attended. Specifically, it is for the covert attention task. So here, for example, there's a task in firstly, there are two blocks. So in one block, we are trying to see like, we're asking the participant to tell the orientation of this red square, which side it is oriented, and for the other block, we're asking the green ones. So when we are attending to the red block and when we're attending to the green block, the side of the screen which is being attended, like the left side or the right side, is being showing the opposite or the contralateral side activity in terms of negative deflection. So the stimulus locations are randomized so that the person is not being attended the stimulus beforehand. So we'll see a larger P1 N1 then rather than the N2PC component. So the stimulus locations are randomized so that the participant doesn't have a predetermined location of the object. And we use fixation because like for the help of peripheral vision, the subject is asked to do the task. Similarly, the red item immediately popped up from the display. So in terms of the color also, it is showing how it is impacting the component, the ERP component in terms of N2PC. So we are seeing the red one has been perceived earlier rather than the green ones. So this is the contralateral minus ipsilateral difference wave. And the N2PC here is delayed by 50 milliseconds in the attention task in terms of green condition related to the red condition. And similarly, once the stimulus appears, it takes about 200 milliseconds for the attention to shift to the target. So here we are seeing like how covertly we shift our attention using this paradigm. And we see there's a negative going wave over the contralateral hemisphere. When we see the collapse to the contralateral target minus ipsilateral target visual field, the component, there's the similar activity, but like for the red ones, there's a earlier processing. And NTPC is also sensitive to the threat images. Here, when the subject is provided a threatful stimulus like the dog barking or a spoon, the threatful stimulus will have a larger amplitude NTPC curve. So these small sensitive physical properties are being moderated by these components. We are seeing the voltage was more negative, contralateral to the threat than ipsilateral to the threat images. Then we have the contralateral delay activity, which is specifically for the working memory task. Here what happens, this fixation, and we have one side we have the rectangle with the rectangles of different colors and the circles. So these bars and these circles are being presented to the subject, and the subject are asked to like see to maintain these colors, to keep these colors in the memory, to check if this sample array is matching the test array or not, or if there is any difference between them or not. But for these bars, they are of their color are similar. This doesn't change. And we see a similar activity in terms of contralaterally for the working memory task. So initially we see an NTPC component because there's a covert attention towards it. And also this is randomly done because if we see if there's a greater presentation of circles on the right side of the screen, then there will be a larger left hemisphere negativity. So this might be a biased results then. That's why we need random presentation of the stimulus. But after NTPC, we see the contralateral delay activity, which is like the sustained voltage over the hemisphere contralateral to the objects being maintained in the memory. So if the object is being presented on the right hemisphere in the memory, then the opposite, that is the contralateral hemisphere, will be showing activation in terms of the negative positive voltage. And these CDA is observed when there are stimulus. After the presentation of the stimulus, we see these CDA. But in NTPC, we see those stimulus when the stimulus is actually present. So NTPC is for when the stimulus is actually present, and CDA is like after the stimulus isn't presented and the next test is shown. That period we see the contralateral delay activity. That is the maintenance for the working memory task. Then there's a N400. So for N400 we mean like it is for the language component. So here so we are seeing like how much time the 400 milliseconds is being taken to understand like how if there is any violation of grammatical or syntactic violation of any sentence or object, like if you pair an object like there's a nurse dog, so this will have a higher ERP component, higher N400 component, negative P component at around 400 to 600 milliseconds. So similarly, these are for like language related components. For example, I take my coffee with cream and sugar. For example, if we give sugar, then it will be like relatively shorter because we already know the combination. But if we put sugar with cream and dog, it's like a sudden violation of the grammatical sentence. So it will show a greater peak due to negative component. Then we have late positive potential. So these are like observed after the N400, as in like after 400 milliseconds, around that time, 400 to 600 milliseconds. And these are observed when the participant is seeing those stimulus which arouses them. For example, in terms of emotions, emotional tasks, we see LPP specifically. For example, if there are three stimulus, a neutral stimulus, a happy stimulus, and stimulus which is like unpleasant, arousing stimulus. So this rotten meat is shown greater amplitude, greater late positive potential, not reflecting that the potential is being affected by the valence, affected by the arousing rather than valence. So these are small things which affects the LPP. Similarly, yeah. So the arousal here, like emotionally arousing stimulus, we can cross match with the help of electrophysiological, like can use GSR or like can use the EMG. We can see with the help of arousing stimulus that if the person was more aroused at that point, so we can measure through LLP also. And this is like lateralized readiness potential. So when we have a task and there's a motor task, for example, there's a stimulus and there's a two stimulus, that is one is we need to press a button if we see a odd number, and one button for when we see an even number. So the object which is there, the lateralized lateralized hemisphere will show the activity. For example, like if we see the object on the left side of the screen and we need to press a right button, the left hemisphere will get activated. So like the lateralized, same as the kind of NTPC, like that contralateral activation is being seen, but the task is the motor task here. Similar in terms of left and right hemisphere, like if the left hand ERP is there, we will see a right hemisphere with greater contralateral activity in the right hemisphere. If the right hand right hand is being pressed in response for response, then we'll see a left hemisphere contralateral activity. So this is another ERP component, which is like the distractor positivity. So what is distractor positivity is like similar to NTPC. So this component was like explored by Rita Sawatsky in the Steve Lux lab, a postdoc. So initially she was trying to study the idea of singleton. Singleton is like the objects which like instantly causes our attention to catch our attention. For example, if there's a bunch of white squares and if I present a red square, so in spite of not telling like our brain will catch that red stimulus, like the red square. So for this way they designed the task, and they were trying to see like the role of those singleton, those distracting stimulus. So while studying this NTPC, they observed this distractor positivity, similar to NTPC, but in the terms of positive polarity. So distractor positivity is kind of plays role as an inhibitory process when we're actively trying to inhibit a particular response and don't attend to the task, as in don't respond to the task, we see those destructor positivity, which is like a positive voltage over the hemisphere contralateral to the destructor. And these are lateralized to the destructor region. For example, color pop out and object pop out. So that is why. And this is observed in around 100 to 400 milliseconds. Next comes the event-related negativity, which is like, so if you observe event-related negativity when the person is trying to understand the task and when there are error present, we are trying to understand the error and we are like constantly manipulating and monitoring the errors. So we see greater, larger negative amplitude at the region, at negative polarity at around 100 to 300 milliseconds. Specifically for the incorrect response, we are seeing the greater amplitude, greater negative component, because like it is like kind of error detection monitoring. Specifically, it comes from the region of anterior cingulate cortex. So we are seeing that in error-related negativity, and yeah, the cortical generator is the dorsal portion of the anterior cingulate cortex. Now we'll move on to the ERPs in the clinical setting. So in this component, the contingent negative variation, we see, firstly, what is contingent negative variation? Contingent negative variation is a kind of component where like it sees the expectancy of the task, as in like when we are presenting a stimulus, I need to, for example, the similar task in the odd and even number button pressing. So I know the next number might come an odd or next number might come an even if I know that. So there will be an expectancy of that object coming. So there will be a pre-stimulus activity of that task. So that is being monitored by the contingent negative variation, which is like the kind of early component only. So in various diseases, these CNV variations has been observed in terms of amplitude. So in Huntington's disease, we see a reduced amplitude, CNV amplitude, and this amplitude reduction is being correlated with the bradykinesia scores of their like high bradykinesia, low bradykinesia scores, and the basal ganglia dysfunction, because in terms of motor response, there's a dysfunctioning. The also in multiple sclerosis where there's like a white matter fabrication problem, myelination problem. So there's a reduced amplitude of contingent negative variation. In mismatch negativity, we were seeing that various elderly subjects, like healthy adults, there is a reduction in the amplitude of MMN in terms of auditory MMN and delay in latency, because the processing perception of the stimulus causes them based on the gyrification of the cortex. So it reduces the latency. Also in terms of various stroke patients and various Parkinson's disease also, people have shown like there's a reduced amplitude and reduced latency. But in some specific disorders like autism spectrum disorder or obsessive compulsive disorder, we see an increase in amplitude of mismatch negativity. This is because like they're more attentive towards the task or greater processing. So here, mismatch negativity is a very effective ERP component when we are using it for infants, like when we are studying the auditory brainstem responses. So these are very fast auditory brainstem responses can capture within infants. Like it's very faster than the visual system for both adults and infants. So we can study infant population from with the help of these components, infants. And also we can study the coma patients, because they're not able to identify the objects and stimulus. But when we give a specific tone and with the help of standard and deviant tones, we can see this particular waveform, the standard minus deviant waveform, and we can predict like, yeah, the person is understanding, their brain is still working. So with the help of this ERP component, we can study these areas. And P300 amplitude, we see there's a decreased amplitude in schizophrenia. So various researchers have used P300 amplitude in among schizophrenic patients. There's a longer latency speed in pre-300 in terms of demyelination. And dementia patients also have an increased latency, as in they require greater amount of time to process to that, categorize that stimulus. We can see here that the same oddball paradigm, if we give it to the patients with schizophrenia or people with schizophrenia, there are like similar activities, like similar time it takes, but the amplitude is similar, but the mean reaction time there's a difference between that. There's a lag. So what is it which is causing this delay in these people? So with the help of this component, they have determined that this might be the post categorization slowing. It is not the perception which might be delayed, but it is the post categorization slowing. So it might happen for this. Sorry. Yeah. So here, like we can see similar actuation, like similar amplitude, but the problem is in the post categorization slowing, and we can see observed through rare minus frequent difference wave. And if we say like in which areas the person is fine, for example, N2PC. So we have a similar configuration of controls in people with schizophrenia in N2PC in the terms of covert shifting attention. So this is like contralateral minus accelerated difference waveform, and we can see like similar orientation. So I will take a short break and we'll see the video, and we'll come back sometime. I'll share the screen of how our ERPs and what does ScienceBeam does. We'll just share.